All right, hey everybody. So we're moving on now to our looking at karyotypes and chromosomal mutations. So let's get right into it. So here we're going to look at chromosomes are going to be, when we're looking at karyotypes, the chromosomes are going to be matched together in homologous pairs. So homologous just means basically that they're the same chromosome, they're the same size, they have the same genetic information on them, the centromere position is the same. So, and you can see them right here. So this is an example of homologous chromosomes. You get one homologous chromosome from your dad, right? Here's paternal. And then we get one from mom, which is our maternal. So you can see that they're the same size, their centromere position is the same up here, and the gene locations are the same. So you can see this codes for the same gene, codes for the same gene, right? Same gene, same gene. What's interesting though is that even though they code for the same gene, it doesn't necessarily mean that they have the same exact alleles. So even though, let's just say this coded for, I don't know, color of your hair, let's just say, which it doesn't, but this codes for color of hair. So, dad gave his gene, his allele for color of hair, mom gave her allele for color of hair, but they were different. So let's say mom had blonde hair and dad has brown hair, so they have slightly different alleles, but both code for hair color. And so that's what we look at when we're looking at homologous chromosomes. Some might be exactly the same, some might be slightly different, depending on which parent gave what. And here's just another image just to show that we have our homologous pairs. We have mom and we have dad, right? Mom and dad. So they both give a chromosome and they're both same size, contain the same genetic information, but they might have slight variations of the alleles. So here we're looking, this is a karyotype. We've talked a little bit about karyotypes already, but as a refresher, the karyotype is when we have our chromosomes are set up in this way, so it makes it easier for us to identify our homologous chromosomes. So, number one, here's our first set of homologous chromosomes, here's our second set of homologous chromosomes, and so on. When we're looking at heterozygous chromosomes, those are like if we looked at number 1 and number 2 and put them next to each other, those are heterozygous because they're different from each other. They don't have the same genes, they're not the same size, so that's what we say as heterozygous chromosomes. So when we look at this karyotype, we will notice that we're going to have these different pairs. And for the different pairs, these are called our autosomal chromosomes. It goes all the way through 1 through 22 are your autosomes. And so those are going to control things that are not specifically linked to your if you're going to be a female or if you're going to be a male. Those are other traits like, the development of your skin, the development of heart, development of what color hair you're going to have, different things like that are going to be part of your autosomes. And then for your last pair, it's not labeled, but it's like our 23rd pair, I guess, but they're not exactly the same, could be your X and your Y. So, and this is showing a male because it has the Y chromosome. If it was a female, it'd have 2 X's next to each other. So this is just showing some examples of different organisms and the pairs that they have. So humans, we have 23 pairs, which gives us a total of 46 chromosomes. Dogs actually have more pairs, cattle have more. So you can just see these different amounts of genetic material. What's interesting is apes, which is has a common ancestor with humans, have a very similar number of pairs. So we have a very similar number of chromosomes. And here we talked about this, but once again, a refresher. We have our autosomes are 1 through 22, and then we have our sex chromosomes, which will determine an individual sex, and they're down here. So females are XX and males are an XY. If we look at this, I want you to try to figure out, which karyotype set is showing the correct number of chromosomes. So take a minute and try to think about, which one's actually showing the correct number that it should have for a human. So what we'll see is that this karyotype A is a normal human, doesn't have any abnormalities in the sense it doesn't have an extra chromosome or anything like that. This one, karyotype B, if we look through, usually for humans we have 2 sets, right? One from mom, one from dad. But if we look at 18, we'll see that we actually have more. We have, instead of having 2, we have 3. So maybe mom gave 2, or maybe dad gave 2. And so that was just something to recognize, that that's an abnormal number of chromosomes. And also to recognize that this is a female and that this one is a male because it has the X and the Y. So what can result, or why can this abnormal number of chromosomes happen? Well, one of the major ways that it could occur is something called non-disjunction. Now, we haven't talked about meiosis yet. Well, we will really soon. But basically, meiosis is very similar to mitosis in that our sister chromatids need to line up in the center of the cell, and then they get pulled apart. Same thing happened in mitosis, right? During metaphase, those sister chromatids, and you can see it right here, they line up in the center of the cell. Those spindle fibers are microtubules. They're going to pull the sister chromatids apart, right? We see them pulling apart. These top 2 are showing proper non-disjunction, or proper disjunction, because they're separating correctly. You know, half is going to one side of the cell, half is going to the other side. But this is showing improper disjunction, which we call non-disjunction, where now the sister chromatids didn't actually separate. So one cell will have too much genetic information. So that could result in, in the end, if that cell gets fertilized, let's just say, now that cell has too much genetic material. And we can see it here. For meiosis, there's actually 2 parts. You have meiosis 1 and we have meiosis 2. And we will go over those in the next video, actually. But non-disjunction can happen either during meiosis 1 or it can happen during meiosis 2. But this is an example of it happening during meiosis 1. If it happens in meiosis 1, that's a bigger issue because now all of the cells don't have the correct number of chromosomes. In this particular example, the correct number of chromosomes would be 2. But you can see that this one has 3, this one has 3, this one has 1, this one has 1. So all are abnormal. So if non-disjunction happens in meiosis 2, so we can see here it was normal. They separated correctly. But if it happens in meiosis 2, non-disjunction will result here, and what we'll see is that 2 out of the 4 final cells are affected and they have a different number of chromosomes. So this one has 3, this one has 2, this one has 1, but what we really wanted was for them to have 2. But not as bad though as compared to non-disjunction happening in meiosis 1. So what we want to look at now is we're going to look at, okay, well, what happens if an individual does receive an abnormal number of chromosomes? We'll look at how if you receive an abnormal number of sex chromosomes, how does that affect an individual versus what happens if you have an abnormal number of autosomes. Typically, if you have an abnormal number of sex chromosomes, it tends to be a lot less severe, maybe because the size of the Y chromosome is so small, potentially, and also because of X chromosome inactivation. So if you have 2 X chromosomes and one's not good, let's just say, then the other one will kind of pick up the slack. But for autosomes, you don't have that kind of luxury. So let's look at some examples. So here's our first one. This is called Klinefelter syndrome. This is when an individual inherits 2 X chromosomes, right? Most people only inherit 1 X chromosome if you're a male. So this individual inherits 2 X chromosomes and a Y chromosome. So because this individual inherited a Y chromosome, then it's going to be a male. It has to be a male. And these are just some of the characteristics that are associated with Klinefelter syndrome. Still can live a fairly normal life. Brain development is good, so they're intelligent in that sense. So there's no abnormalities like that. But these are some of the characteristics that are associated with Klinefelters. Taller, less muscular body than males their age. They tend to have broader hips and longer legs. They have excess skin up here, weaker bones, lower energy. They might have a delay in puberty. They tend to have a lot less facial and body hair. So those are just some characteristics that are associated with Klinefelters. But sometimes you wouldn't even know. You wouldn't even notice that somebody has Klinefelter syndrome. Turner syndrome now is actually when an individual only inherits 1 X chromosome. So it's still a female, but you can see with this karyotype, all of these were normal except they only got 1 X chromosome. So here are some of the characteristics that are associated with Turner syndrome. So sex chromosome abnormality, like we just talked about, they're typically much shorter in height. They have like this webbed neck, excess skin on the side. There's also some issues with their kidneys, skeletal/bone abnormalities, and loss of ovarian use. So they are infertile, so they cannot have children. Now we start to get into some chromosomal abnormalities where we look at the autosome. So those two examples were of your sex chromosomes, but now we'll look at, I think we have 2 or 3 examples that look at autosomal abnormalities. So for this one, trisomy 21 is known as Down syndrome. Maybe you've heard of that before, but what that really is showing is that when we're looking at our chromosomes, if we look at chromosome set number 21, instead of only having 2 like all these other ones, there's actually 3 right there. So we call it trisomy because trisomy means 3. So there's 3 chromosomes at chromosome set 21. But everything else is the normal number of chromosomes. So we see here, this is one of the more common chromosomal abnormalities. Then we also see some symptoms: mental retardation, characteristic facial features, short stature, tend to have heart defects, and a shortened lifespan. This kind of characteristic of the Down syndrome tends to increase with the mother's age. So the older the mother is, the more likely this is. It's not always the case, but it just seems to be a bit of a pattern that has emerged. But the person can live with this. They can live with this and live a very happy life with this. But this one is not good. Trisomy 18, this is Edwards syndrome. So this is when an individual has 3 chromosomes at 18. This leads to a lot of issues. So if the baby is even born, it might not even survive to birth. But this is autosomal. This is going to be where we have a really small heart develops, abnormally shaped head, mental retardation, clenched fists. I mean, most babies do clench their fists, but they like really clench them and they're like overlapping their fingers. Really low birth weight, and they usually will die before birth or after, and they might only live a year. So you can see that having an abnormality in your autosomes is a really big issue, and most of the time the individual cannot survive. It's just way too much genetic material. With Down syndrome, that was trisomy 21. So you can see that these chromosomes are much smaller, which might be why a person with Down syndrome can live a more normal life and survive for much longer, as opposed to 18, which those are huge chromosomes, and they contain a lot of information. All right, our next one. Here's Patau syndrome, another really severe genetic disorder where we have, this is actually when we have 3 chromosomes at chromosome number 13. So trisomy 13 it's called. So it's characterized by severe intellectual disability, heart, brain, and spinal defects, a cleft lip or palate, and that's what it would look like right there. It is a life-threatening condition. It's fatal, and the baby could not live a very long life at all, unfortunately. So you can just see that having the autosomal disorder is much more severe than having a sex chromosome abnormality. All right, so really the last thing that we're going to look at is just not necessarily the number of chromosomes is different, it's just there's some changes in the actual chromosome itself. So let's actually get right into it. Here are the definitions of each one that we will discuss. We have a deletion, where we see if this is our chromosome, a piece of the genetic material is missing. A duplication is like, if this was the normal one, it actually has now 2 of the same set, which might not be a problem. And then inversion, we have the reversal of a chromosomal segment. So you can see right here that they've been switched. And then translocation is actually when one piece of a chromosome from one chromosome switches with a completely different chromosome. So they've switched places. And so we're going to look at one example. This is reciprocal translocation. And this is a form of leukemia. Leukemia is associated with blood disorders. So in this particular case, we have chromosome number 9 and we have chromosome 22, And this piece of chromosome 9 is actually replacing chromosome number 22 at that point. And then the same thing happens with chromosome 22 replaces this section. So we can see that those traits, those genes have been like swapped to different chromosomes. And because of that, that actually affects the number of white blood cells. So now there's way too many white blood cells in the blood, and that actually blocks healthy red blood cells and your healthy platelets. They're being they're being bombarded by all these white blood cells. There's just too many in there, and your body can't function well. So, all right, so that is the end of our notes for looking at our mutations and looking at some chromosomal abnormalities. All right, I hope it was helpful, and if you have any questions, just let us know. All right, bye.